So today we have Dr. Kevin Mann from the Division of Molecular Immunology, and he will be presenting T cells and immunity to viruses.
Okay, good morning everyone. So today I'll just be talking about our work we do on T cells and how they're important for our immune system to combat viruses. So just quickly, T cells are important for a number of functions, including killing cells that in your body that are infected with bacteria or viruses. And they also help other cells in the immune system. And work from our group and other labs have also shown that T cells are important for detecting tumours in your body and killing tumours. So they're important in the context of cancer. So this is just a simple explanation of how T cells respond to a pathogen that you encounter in your body. So within your blood and in your organs, especially in your lymphoid organs, such as your spleen or lymph nodes, you have naive T cells that circulate around your body. And they come into contact with antigen-presenting cells, which is shown here in this, which is a dendritic cell. And these are the cells that present parts of bacteria or viruses that are present in your body to the T cells. And certain T cells will recognize these bacterial or viral components and will undergo a process of activation. So the cells become larger and then they start dividing. And then they undergo a process known as differentiation and massive expansion. So during an infection, the number of T cells in your body expand about anywhere from 1,000 to 1,000,000-fold. And then once the T cells have killed all the infected cells in your body, and including any other viral particles that may be present, they undergo death. So most of them die. And then a few will remain that memory cells that stay in circulation for a number of years. And memory cells are important because they provide quicker protection for you when you, say, have a cold, or if you get the flu a second time, you'll recover a lot quicker. So this is just what a normal or a typical T cell response looks like. And over here you have the number of days after an infection, and this is the population size on the y-axis. So when you get a virus or a viral infection, typically the virus replicates very quickly, and your T cells will take a number of days to divide and expand to catch up. But once they reach a certain number, they're able to kill all the infected cells very quickly. And so the virus goes down very quickly, and then your T cells will start to die, and then this is the memory population pool. So typically, a response to the virus doesn't last more than 1 or 2 weeks. And the type of cell that I work on in particular are cells called CD8 positive T cells, and they are known as killer T cells. So these cells are the ones that kill other infected cells in your body. And they do so via a number of mechanisms. And they have to first be able to recognize a particular part of the virus or bacteria before they do so. So this prevents T cells from killing non-infected cells in your body. So this is known as antigen recognition. And when this occurs, so when the T cell recognizes an infected cell, then they form a contact with the infected cell, shown here, and then they release components into the infected cell to mediate cell death. And these are known as granzymes, which activate things called caspases in the cell. And this causes the cell to die, which is shown here. And when the cells die, they undergo shape changes, so they look like that. And also in our lab, we work on proteins called transcription factors. I know Renee probably would have explained briefly what transcription factors are. And basically, transcription factors are proteins that bind to DNA at certain sequences and they regulate the production of other proteins from other genes. So for example, here we have, we call this transcription factor A, which binds to a segment of DNA before this gene X. And this binding will promote the production of something called RNA, which is basically a copy of the DNA sequence. And then from this RNA, it's actually a transcript for a particular protein to be produced. And also what Renee probably would have mentioned is that transcription factors themselves are created via this process. So factor A would have been transcribed and translated in this process. So some of the transcription factors we work on, or proteins, are IRF4 and BLIMP1. And the people that work in mouse rooms 10 and 11 may have heard of these knockout mice or may take care of these mice. And so what we found is these two proteins are very important for T cells to expand when they encounter a pathogen, and also for them to survive during this response. And some other transcription factors you may have heard of that our lab work on, ID2, ID3, BAC2, BATF, and E2A. So these are some of the mouse lines that we have. So I work in particular on the protein called IRF4. And what we found out is it's critically important for T cells to survive and for them to expand properly during infection. And this graph is a very simple experiment where we just take T cells out of the mouse, out of the spleen, and we compare a wild-type mouse and an IRF4 knockout mouse. And so we place these T cells that we isolate from the spleen into a Petri dish and grow them in an incubator for a certain number of time. So the time is on your x-axis there. And then we count the number of cells after we stimulate them to start dividing. And as you can see in the black bars are wild-type T cells that grow and expand in numbers over time. And you can see that when you don't have IRF4, you have much lower numbers of T cells. So then we try to find out whether these proteins are important inside a mouse or in humans during a response to a virus infection. And two such models we use are influenza, or LCMV, which is also known as lymphocytic choriomeningitis virus. And these are two common viruses we use to look at T cell responses in mice. So a very simple experiment we can do is to take a wild-type Black6 mouse and compare them to whatever knockout or gene you're interested in. And in this case, I've just looked at an IRF4 knockout mouse. And so then we take these mice and infect them either with flu, which is an intranasal infection, or LCMV intravenously. And we take organs at certain times after infection, and then we look at the cells in the organs to see how they respond to the virus. And one technique that most immunologists use is called flow cytometry. And this is a method we use commonly to detect certain cell populations within organs. So very simply, I know this is probably a very complicated diagram, but what we do is first we get cells from either blood or certain organs, and you can do this with human samples too. And then we stain these samples with fluorescently labeled antibodies. So these antibodies will fluoresce if they get excited by a certain wavelength of light. And we have machines that have lasers that emit light of certain colors. And so we pass our cell samples through these machines that then will pass through a laser beam, and they'll get these fluorescently labeled antibodies will get excited by this light and emit light of a certain color. And this color that's emitted will be detected by a detector, and then we can isolate different populations of cells And typically the data looks something like this. So we can detect different populations of T cells or B cells or whatever cell you're looking at by this method. So this is what we do to look at T cells that respond to certain viral infections in our different knockout mouse models. So in this experiment, I've just done a flu infection of wild type and RF4 deficient mice. And this is taking spleen cells at day 10 after infection. And using this flow cytometry method, we're able to detect T cells here that specifically recognize a part of the flu virus. And this part of the flu virus is called MP366, which is a nuclear protein. It's just part of the viral component of influenza. And as you can see here in the wild type mice, you get a nice population of cells that can recognize the virus. But when you don't have RF4, you don't get cells that recognize the virus. And this is just the percentage of cells that are virus specific. And another way we can look at whether T cells are able to combat viral infections properly is to do something called a viral plaque assay. And this is what we do to look at whether T cells can respond properly to viruses such as LCMV. And very simply in this assay, we take your infected mouse spleen or whatever organ you're looking at and mash the organ up. And then we dilute the organ out a number of times. So we do something called a serial dilution. And then we plate the diluted virus stock out onto Petri dishes that contain agar and other cells. And then we measure how the virus grows on these cells. And typically, when virus infects a cell, they lyse the cell and will spread to surrounding cells and will form something called a plaque, which is a spot on the layer of cells here. And so over time, you'll see if you have a lot of virus in your sample or in your organ, then you'll get a lot of these plaques forming. And then you can take these dishes and count them and determine how much virus concentration you have in a certain organ. So this is what we've done with two mouse lines that we have called the IRF4 Flox CD4 Cree mice. So in this experiment, we've just taken a wild type mouse that doesn't have CD4 Cree and a mouse that does have CD4 Cree. And this CD4 Cree mouse just means that IRF4 is only missing in T cells, but other cells are normal. So then we infect these two mice and take organs from the spleen 30 days or how many days you're looking after infection, and then we measure the virus concentration in different organs just to see whether there's differences in these two mice to be able to combat or control infection over time. And these pictures on your left are a typical picture of what a viral plaque assay would look like. So once you've done this assay, then you count the number of plaques to determine your overall concentration of virus in your organ. So here I've just done spleen and kidney. And this graph just shows that the wild type mice are okay at controlling virus over time. Whereas if you knock out IRF4 and T cells, then you have much higher levels of virus in the spleen and kidney, meaning that IRF4 is very important for T cells to control virus infection. And so next we want to know what kind of genes IRF4 controls in the mouse or in the immune system. So we have to ask, why is IRF4 important? So we have to ask what kind of genes or proteins does IRF4 control within the genome of the mouse. And one method that I think Renee also explained earlier is we use a method called Chromatin Immunoprecipitation. It's quite a complicated process, but I think it's quite easy to understand. So, as I explained to you before, IRF4 is a transcription factor protein that binds to DNA. So, in order to find what genes IRF4 binds to within your whole genome, we take cells. In this case, I'm interested in T cells, so we take all the T cells out of a spleen, and then we do something called cross-linking, which binds the protein to the DNA. And then we subject the cell samples to fragmentation using high-frequency sound waves. So this breaks up the DNA into small fragments. And then we use antibodies. So these antibodies bind to the protein specifically. So we're only using antibodies against IRF4. So other proteins that are bound to DNA won't be captured in this process. And then we remove all the other proteins and antibodies. And what you're left with are the sequences of DNA that are specific for IRF4. And so your DNA code is made up of 4 base pairs, A, C, T, and G. And these repeat in multiple different orders. And so typically a fragment of DNA, when you sequence it, will just have a string of letters that look like that. So when we sequence these bits of DNA, we get a string of letters that are maybe 100, 200, or 500 letters long. And then we have to match it to your whole DNA sequence, the genome. So we have people that do that in the bioinformatics division. And typically when we get the data back, we get something that looks like this. So this is the BLIMP1 gene. And if you can see here, this cone-looking thing here is actually fragments of DNA on top of each other. And they create, when they stack up on top of each other, they create this peak. So we know for sure that IRF4 will bind to that sequence of DNA. And also, we know that it's important for producing BLIMP1 protein. So in this process, we found that IRF4 binds to thousands and thousands of genes that are important for T cells. And some of the most important genes were the genes that were involved in how cells use glucose. So like us, T cells like something sweet every now and then. And for this process of division and expansion, they actually need a lot of glucose, because this process is very energy dependent. For them to divide, they have to create essentially all the components of themselves again. So we found during this process of chromatin immunoprecipitation and other experiments that IRF4 controls a lot of genes involved in how cells use sugar to divide. And this is just a simple experiment we've done where we've taken glucose that has a fluorescent label on it. And this just measures how much sugar the cells take up over time. And as you can see here, the wild type cells take up quite a lot of sugar over time. And if you don't have IRF4, then they take up lower amounts of sugar. So they're not able to have enough energy to divide and expand like normal cells do. So I'll just leave it there, and I'd just like to thank everyone from the Callie's lab and the molecular immunology division and all the past and present animal technicians that have been critical in doing this work. Of course, these include people in the quarantine facility and also at level 4 at the moment. So thank you very much for your attention. Yes?
With those dishes, when you're counting the plaques, I noticed one of them is with really high burden. How do you count all of those?
Oh, okay. So I'll just go back.
Yeah, like that top left one. How do you count all of those?
So Because we've done the dilution, if it's too much for you to count, then you'll count the one that's more dilute, and then you multiply it back to find out like your total overall concentration. Because sometimes when you do these types of experiments, you don't know whether you're going to be able to sit there and count like a million little spots there and get it right. So that's why we do different dilutions out. And if it's too concentrated, you do the next one down, and then you can work back just by multiplying by 10 or however many you've diluted it by, what your concentration is.
Thank you very much.
Some chocolate mice.
All right, thank you.
Thanks, Kate.